Welcome to the lecture of Plant Molecular Biology on phytochemicals from plants, biosynthesis, and functions. The sum of all of the chemical reactions that take place in an organism is called metabolism. Most of that carbon, nitrogen, and energy ends up in molecules that are common to all cells and are required for the proper functioning of cells and organisms. These molecules, such as amino acids, carbohydrates, nucleotides, and lipids, are called primary metabolites. Unlike animals, however, most plants divert a significant proportion of assimilated carbon and energy to the synthesis of organic molecules that may have no obvious role in normal cell function. These molecules are known as secondary metabolites. Secondary metabolites generally, but not always, occur in relatively low quantities, and their production may be widespread or restricted to particular families, genera, or even species. The distinction between primary and secondary metabolites is not always easily made. At the biosynthetic level, primary and secondary metabolites share many of same intermediates and are derived from the same core metabolic pathways. For many years, the adaptive significance of most secondary metabolites was unknown. These compounds were thought to be simply functionless end products of metabolism, or metabolic waste. Today, we know that many secondary metabolites have important ecological functions in plants. They protect plants against being eaten by herbivores and against being infected by microbial pathogens. They serve as attractants such as odor, color, and taste for pollinators and seed-dispersing animals. They function as agents of plant-plant competition and plant-microbe symbiosis. The ability of plants to compete and survive is therefore profoundly affected by the ecological functions of their secondary metabolites. Secondary metabolites are molecules that are not essential for the organisms producing them, but are only produced during distinct phases of the life cycle or under certain environmental conditions, such as induced by attacking pathogen or treatment with inducing agents. These metabolites arise from a number of biochemical pathways and comprise a wide variety of chemical structures. They are commonly classified according to their precursor molecules from primary metabolism and the enzyme class that synthesize their basic chemical structure. Plant secondary metabolites can be divided into three chemically distinct groups: terpenes, phenolics, and nitrogen-containing compounds. The terpenes or terpenoids constitute the largest class of secondary metabolites. Most of the diverse substances of this class are insoluble in water. The terpenes are classified by the number of C5 units they contain, and they are synthesized through two major pathways: the mevalonate pathway and the methyl erythritol phosphate pathway. Isopentenyl diphosphate is the building block of terpenes and combined with its isomer dimethyl allyl pyrophosphate to form larger terpenes and isoprenoids, a diverse class of over 30,000 biomolecules such as cholesterol, vitamin K, coenzyme Q10, and all steroid hormones. Certain terpenes have well-characterized functions in plant growth or development, and so can be considered primary rather than secondary metabolites. For example, the gibberellin, an important group of plant hormones, are diterpenes. Brasinosteroids, another class of plant hormones with growth-regulating functions, originate from triterpene. Sterols are essential components of cell membrane and are derived from triterpenes. The red, orange, and yellow carotenoids are tetraterpenes that function as accessory pigments in photosynthesis. Abscisic acid is the sesquiterpenes produced by degradation of carotenoid precursor and functions in many plant developmental processes, including bud dormancy and stress responses. The vast majority of terpenes, however, are secondary metabolites presumed to be involved in plant defenses. Terpenes are toxins and feeding deterrents to many herbivores, insects, and mammals. Thus, they appear to play important defensive roles in the plant kingdom. For example, monoterpene esters called pyrethroids found in the leaf and flowers of chrysanthemum species show striking insecticidal activity. Both natural and synthetic pyrethroids are popular ingredients in commercial insecticides because of their low persistence in the environment and their negligible toxicity to mammals. In conifers such as pine and fir, monoterpenes accumulate in resin ducts found in the needles, twigs, and trunk. These compounds are toxic to numerous insects, including bark beetles, which are serious pests of conifer species throughout the world. Many plants contain mixtures of volatile monoterpenes and sesquiterpenes, or essential oils, that lend a characteristic odor to their foliage. Peppermint, lemon, and basil are examples of plants that contain essential oils. Essential oils have well-known insect repellent properties,